we kind of officially get started, and I hand it over to Jake to introduce our speaker. Just one quick announcement. We have one more speaker, part of our spring seminar series. So next week, right back here in this room, Seth Davis in the Department of Forest, Range, and Stewardship will be talking about bees and the flowers they visit in the Colorado Front Range. So hopefully we see you back here next week for that. And without any further ado, I'll hand it over to Jake to introduce our speaker.
Okay, thanks, Troy. So if you don't know me, my name is Jake Horkamp. I'm a research scientist. I work in the Restoration Ecology Lab with Mark. So it's my honor today to introduce Professor of Restoration Ecology, Mark Paschke, for this seminar. When Troy asked me to introduce Mark, I knew I had to tell a good Mark story, right? And so I was thinking about it the last couple of days. And way back when I was just sort of getting started working on my Ph.D., I was up in Wyoming. I was setting up some research plots, and they were pretty big plots. And I was out there like the graduate student I was trying to like line up the edges of the plots with a compass and like square off the corners and figure out how to get this perfect rectangle. And my advisor at the time, Paul Nyman, came over. He finally caught up with me. He explained to me that I could use the Pythagorean theorem to just sort of lay out like a perfectly rectangular plot, right? And at the time, it was like this very rare and poignant example of the relevance of high school math. But fast forward a few years, working in the lab with Mark, we're out at Waverly, setting up a new study. I think it was Haley's study. And I got there a little early, so I got started, and Mark arrives, and I can just tell the way he's kind of walking over. He's like 112% sure I'm doing something wrong. And I'll just be honest with you that I was definitely not setting things up in the correct way. But one thing I was doing right is I was using the Pythagorean theorem to lay out a perfectly rectangular plot, right? And I explained to Mark, I was like, that's how Myman showed me how to do it way back in Wyoming. And Mark was like, well, I showed Myman how to do that. And Ed Redente showed me how to do that. And Chuck Bonham showed Ed Redente how to do that, right? And the names get a little fuzzy after Ed Redente, so don't necessarily quote me on that. My point in telling this story is not to suggest that Mark is old. My point in telling this story is to demonstrate kind of how Mark's work and contributions represent a big part of like the institutional memory of the Department of Forest and Rangeland Stewardship and Restoration Ecology and the Warner College of Natural Resources. So I think if you were to jump 20 or 30 years into the future and you were going to write a history of the Warner College of Natural Resources, there'd probably be a chapter in there that said Mark Paschke and restoration in the Warner College of Natural Resources. So I've gained a lot from working with Mark, and I certainly think I'm not the only person in the room who would say that. And so with that, sort of starting us off, take it away, Mark.
Wow. Thank you, Jake. Appreciate that. So thanks for coming. I was asked by Troy, about a month or two ago to give this talk, and Katie wanted my title and abstract pretty much right away. So I sent in a title and abstract thinking, oh, that sounds pretty cool. And then the other day when I went to put the talk together, I was scratching my head wondering what the heck was I talking about when I sent in that abstract and title. But I'm going to try to do my best and stick to the original title, but I'll probably deviate from the abstract a bit. So what I want to talk to you about is really what I see as an emerging approach to ecological restoration that really focuses on processes over composition. But before I do that and jump in, I just want to give you a little bit of background for some context. And that is that a lot of the restoration techniques and paradigms that we use today were really developed a while ago. So back in the 1930s through the 1980s, from the Dust Bowl to the advent of the SMACRA regulations, the Surface Mining Control and Reclamation Act, which really played a big part in how reclamation and revegetation science was done in this country. And a lot of that focused really just on plant cover and often on using non-native species. And that's really kind of set the stage for where we're at today. And what that has resulted in is a lot of declines in really important species because of the loss of biodiversity that we have noticed with birds and insects. Not a new story for all of you, I'm sure, but it's related to this fact that we've had a decline in plant biodiversity that's resulted in these cascading effects, at least partially. And during that time, we became really proficient at growing huge stands of crested wheatgrass and smooth brome, which were great for stopping the Dust Bowl and doing mined land reclamation. But they resulted in really low diversity plant communities that were not very resilient. And we've realized in recent years that if we want forest and rangeland plant communities that are going to be resilient to fluctuations in productivity that are caused by climate change, we need highly diverse plant communities, which in turn will result in highly diverse insect and bird communities. And so this is a goal that I think everybody is really working towards, is this idea of increasing biodiversity because of the importance that that biodiversity has and is emerging in ecology. And a lot of the reasons why we can't advance, unfortunately, have a lot to do with policy, because a lot of the policies that regulate revegetation specifically really focus on things like percent vegetative cover. So to get a bond released in a mining operation, it often focuses on how much plant cover you have, also how much noxious weeds you might have, or maybe some compositional fidelity to a reference condition. But really not focused on biodiversity. And so what I want to talk today about is shifting that towards this idea of more of a process-based approach to restoration. And one of the problems that we have to overcome if we want to do that is understanding how we can develop policies that focus on process as opposed to form or composition. And I'm going to come back to that at the end of my talk. So just put that in the parking lot for now. So if we want to think about how we want to do restoration better, I think there's broad recognition within the field of restoration ecology that we really need new approaches to the way we go about doing things. And what I want to talk about today is a shift towards a more holistic approach to restoration. One that recognizes that plants have to be considered as diverse communities rather than single species, and that they interact with soils and biotic networks that result in emergent properties. And most of these emergent properties are ones that happen to take place where these three areas intersect, and that's in the rhizosphere of plant roots, where the biotic networks, the functional diversity of plant communities, and resource regimes within soils all come together to really influence what I'm going to refer to as emergent properties throughout the rest of my talk. And if you're not familiar with emergent properties, they're a characteristic, a trait, or a behavior that complex systems possess, which basically means that individual parts of that system do not have their own behaviors, if you will. In other words, the whole is greater than the sum of the parts. And the thing about emergent properties is they're interaction dependent. So they arise from relationships that occur in ecology, things like competition and facilitation, which I'm going to talk a lot about today, and other interactions. They're non-additive. So again, the whole is not just the sum of the parts, and adding species together doesn't linearly predict the outcome of what happens in a community. And they're scale dependent. So things that we observe at higher levels of organization, like at the community, ecosystem, or landscape level, they emerge from mechanisms that operate at much lower scales, like at individual or population scales. So I'm going to talk today about research within 5 general areas that my lab has been involved with over the last 30 plus years here at CSU. So we're going to do some ancient history, as well as some more contemporary history, if you will. And that is focusing on areas such as nutrient cycling, invasion resistance, above below ground coupling of systems, plant community diversity, and long-term legacies of what we do within restoration. And I think our lab has made rather unique contributions to process-based restoration by combining field manipulation of soil nutrients that I'll talk about in a minute, microbial community restructuring, functional seeding optimization, long-term resource trajectories, and applied forest and rangeland management. And hopefully you'll see some intersection of all of those in the projects that I'm going to describe. So I want to give you an overview of what I think of as 8 process-focused research areas that we've been involved with. And I kind of cherry-picked just a few of them. There's a lot more I could have included, but I'll come back to some of the ways these are interrelated at the end. And the first is some really ancient history. It's one of the first projects that I worked on when I came to CSU as a postdoc with Ed Redente and Terry McClendon, way back during the Pleistocene, out on the shortgrass steppe. And of course, the shortgrass steppe looked different back then when I first started working there. But it really focuses on the role of nitrogen availability in plant community assembly. And what we did, this was based on work that Terry McClendon and Ed Redente had been doing over in Western Colorado, where they noticed that nitrogen fertilization had a pretty strong effect on succession in restored plant communities. And so we brought that idea over to the east slope and went out to the shortgrass steppe and set up an experiment in some conservation reserve program lands that had been seeded with similar seed mixes, but they differed in age. So we had one site that we called an early searle site that had been seeded 4 years previously. We had what we called a mid-searle site that had been seeded as part of the CRP program 12 years prior, a late searle site that was 38 years old, and then we had an undisturbed, uncultivated shortgrass steppe site that was located within the CPER out in the Pawnee National Grasslands. And what we did at that site is we put in replicated 10 by 10 meter plots where we either added nitrogen at 100 kilograms of N per hectare per year, we did nothing, or we added sucrose as a way to lower nitrogen availability. And we did that, again, on these areas that had similar seedings. And we did a lot of things to monitor what was happening in these plots. One of the things we did is we monitored nitrogen availability using several techniques. One of them was using ion exchange resin bags buried in the soil that capture ammonium and nitrate over long periods of time with repeated incubations. And what we see is kind of what we would expect in these different sites. When we add nitrogen to the early successional site, we don't see a lot of nitrogen ending up in the resin bags because the plant communities are sucking it all up. These are fast-growing, annual-dominated plant communities that absorb a lot of nitrogen. When we add nitrogen to the uncultivated shortgrass steppe, the nitrogen just kind of sits in the soil because the plants there really don't care. They're growing slow. They don't respond to nitrogen. It stays in the soil solution. We see it in our resin bags. And when we add sucrose, we do indeed reduce nitrogen availability, which was the verification that we were looking for. So let's take a look at the plant community. And this is kind of a wonky way to illustrate the plant community, but hopefully you'll catch on pretty quick. What we have are our 4 different sites and the changes in the plant composition over time. It says, I think the axis says years since last cultivation, but actually it only shows the sampling year. So we're going from the early 90s through the late 90s here. And you can see in the untreated plots, you just see succession proceed from an annual dominated system, which are the yellow shading, to a perennial dominated system in the blue shading at the early successional site. So this is what succession looks like. You go from an annual dominated plant community to a perennial dominated plant community. Things are a little different at the mid-soil site. It was annually dominated for a longer period of time, eventually going to perennial domination. The late soil, mostly perennial dominated. The uncultivated shortgrass steppe changes with time, but generally stays perennial dominated with a pretty good component of succulents, shrubs, lichens, perennial forbs. So what we would expect to see. When we add nitrogen to the system, what we saw was that we essentially increased the abundance of annual forbs, increased their tenure. So even though we stopped our application where you see this line here, we see the annual component extending in time. So we essentially slowed down or changed succession in the system by adding nitrogen. Same thing with the mid-soil. We see a slight response in the later soil and not much of a response in the uncultivated, except during a wet year when we had a flush of native annuals that emerged during that period. When we add sucrose as a negative nitrogen treatment, we see kind of the opposite. We see succession happen faster to a more perennial dominated system in all of the sites with not much of an effect at the late successional site. What's interesting here is, though, in that wet year, we eliminated the flush of native annuals just by adding sucrose, which tells us that those flushes of native annuals that we see in these systems has a lot to do with the effects of moisture on nitrogen availability, as opposed to just the effect of moisture. So nitrogen availability, very important for controlling the successional trajectory at these sites. And related to resistance to invasion as well, that are related to this cycling of nitrogen within the system. So nitrogen availability is really acts as a control knob in these systems on assembly rules, and additions generally increase early successional annuals relative to late soil perennials, while lowering in availability really shifts the balance to the late successional species over the early successional. And by reducing nitrogen availability, especially at that mid-soil stage, we move the plant community towards very quickly a perennial dominated system that resembled the reference condition. So in other words, restoration can be a lot faster when we target the process, in this case, nitrogen cycling, that sets the assembly rule for the way succession proceeds in this plant community. So we continued with this research thread for many years, and it sort of culminated with a review paper that was led by a postdoc in my lab at the time, Laura Perry, who looked at not only our research, but there was a lot of other people jumping into this field at the time, looking at the relationship between nitrogen availability and weed dominance. So Laura reviewed that literature, and there were some conclusions that came out of that, that invasions of annual plants can be controlled by lowering nitrogen availability, and that carbon additions and topsoil removal were sort of the best approaches for doing that. Probably not practical in a lot of cases, but simply illustrating the importance of that assembly rule. And there might be some other approaches that could be used as well. But in general, what the review told us and all the other work that was going on in this field suggested was that by changing nutrient cycling, we could change the competitive rules in the system, which changed the community assembly process. So another research area that emerged from within this, the previous one I just talked about, was an attempt to understand how the soil microbial community was responding to these nitrogen and sucrose additions. So we were doing a lot of side studies in these plots and other plots, trying to understand what was really happening with the microbial community. And we quickly realized, working with Don Klein, who is a professor of microbiology here at CSU, we realized that a lot of the techniques, or most of the techniques for looking at soil fungi, were really inadequate because they kind of were ignoring the networked and dynamic nature of soil fungi. So soil fungi have hyphal structures, some of which are alive and filled with cytoplasm, and the rest of which are just empty dead straws that move material around, both cytoplasm in nutrients. So if they're mycorrhizal fungi that are connected to plants, a lot of that is just dead tubes that are moving stuff around in the system. And we realized by just doing counts of those, we weren't really capturing the dynamics of the system. And so Don Klein developed a technique based on this. But first we published this review paper that sort of talked about this shortcoming in most of the current methods. This idea that most of the fungal hyphae in the soil are these dead tubes that don't contain cytoplasm. Fungi move cytoplasm to areas of active growth where there's a hot spot of nutrient availability, so they can take advantage of soil heterogeneity to capture resources and move them within their hyphal networks. So that review paper led to Don developing a new technique for looking at the microbial community that was based on the idea that filamentous fungi have this variable and changing hyphal network, most of which is devoid of cytoplasm. So we developed a direct integrated measure of the physical characteristics of the fungi and their associated bacteria. And this was a microscopy-based approach that looked at microbial biovolumes, both of bacteria and fungi, the living and the dead components of both of those, and combined them into a single value measure of the microbial community. So very labor intensive, involves staining and looking at lots of slides of soil bacteria and fungi. So we developed this, what Don called the TAF-B biovolume ratio. That was this more integrated measure of the activity of the soil microbial community. And in those plots that we were studying at the shortgrass steppe, this TAF-B ratio did a very good job of correlating with what we were seeing with the plant community and with nutrient cycling. So we thought it was a pretty good measure moving forward to use. Unfortunately, because of the expertise it requires in doing microscopy and staring through a microscope for hours and hours and hours, it never really turned out to be practical for any larger sense, but very interesting idea. And again, it pointed to this notion that it's really the activity and the processes that these organisms are involved with that's more important than their just presence or absence. So moving on, another research area that we moved into happened in the around 2010, 2015 timeframe, when there was a lot of oil and gas development going in out of the shortgrass steppe. And a lot of the regulators and the oil and gas industry people were curious about how best to do revegetation. And most of the revegetation that was happening out there involved using seed mixes of native species that contained usually between 5 and 10 species of native plants. And the question was, why weren't they seeing diversity in their reclaimed areas? And it probably had to do with the lack of diversity in the seed mix, or maybe a lack of number of seeds they were putting on the site. So the oil industry came together and sponsored a study with us looking at how to optimize, or what is the optimum rate at which you should seed restoration sites in the shortgrass steppe, and what's the optimum number of species that you should include in the seed mix. We started to look at different experimental approaches for doing this. And Jane Jonas, who was a postdoc in my lab at the time, came up with a novel approach. And what that involved was looking at these different rates of number of species and number of seeds in the seed mix. So we had 9 different levels, starting with no seeds to different levels of combinations of these. And the reason this was unique is because we stole the technique from the pharmacology industry. So in pharmacology research, they're often interested in what is the optimum interaction dosage between 2 different drugs. And because they're limited by doing research with human subjects, they came up with this novel response surface regression approach, which allows one to look at the interaction between two variables without having lots of replicates involved. And so a response surface regression, what you do is you have a single value repeated for these different combinations of these two variables, in our case, seed mix diversity and seeding rate. And then you have one in the middle of this response surface that you replicate. So we set up replicated blocks where we had one of each of these and then 9 of these. So we had blocks of these replicated at 12 different sites, so 12 different blocks with this. And this allowed us to, rather than having to have 9 replicates of each of these, it allowed us to condense this down into a single, more manageable experiment. And the other thing it does is it allows you to determine a sweet spot, which is what they're interested in. The drug research is finding a sweet spot for the interaction of those two drugs using a limited number of replicates. And so by doing this, again, we used these 12 blocks across 3 locations, and then we looked at the response of the restored plant community based on 9 integrated variables. So things like abundance of native species, a number of non-native species. We combined all of those into a single measure to find that sweet spot. And what we came up with was this was our sweet spot. So a diverse mix of around 40 species seeded at a rate of around 1,000 PLS per square meter. That gave us the most favorable restoration outcomes. So the takeaway from this was that if you want to do something with your seed mixes, increase the diversity and hold the seeding rate high compared to industry standards, but you don't need to go too crazy. And the other thing that came out of this was an observation that a lot of what did very well and drove a lot of our response variables was the inclusion of native annual species within the seed mix, which led to favorable outcomes. So that was research area number 4. So research area number 5 was something that I was very fortunate to have when I came to CSU. I kind of inherited a large research project site over in Western Colorado that had been put in back when I was in elementary school, back in the 1970s and early 1980s. And this was put in by Ed Redente and some other range professors back at that time. At the time, there was a lot of oil shale activity supposedly going to happen in Northwest Colorado, lots of money flowing in to look at ways to restore areas that might be impacted by really heavy oil shale extraction in Northwest Colorado. And so CSU set up this long-term study site in the Piceance Basin where they put in all kinds of experiments. I believe there's 12 different studies, probably more than that actually, probably like 15 or 20 studies that were put in this permanent research place. And I inherited these studies, which was awesome because long-term revegetation studies are hard to pull off because it takes a long time and a lot of funding. But to be given a site where different things had been done for revegetation back in the 1970s and 80s, it was really a wealth of information that we could pull out of that site regarding long-term outcomes and what really matters and doesn't matter in terms of plant community assembly. So one of those studies was led by Tim Hosel, a graduate student in my lab, looking at a study where they seeded different seed mixes on plots in 1984. So in some plots, they seeded an early successional seed mix that included both native and introduced species. This is the early 1980s when people didn't care about seeding non-native species as much as we did today. But there was both native and introduced included in these seed mixes. a late cereal seed mix, again, native introduced, and an unseeded control. And we went back to these study plots when Tim was a graduate student many years later, 25 years after these plots were put in, and looked at the long-term outcomes. And what we found was that these different restoration seed mixes resulted in very different plant communities after 25 years. The early successional seed mix was no different than the control, where there was no seeds added because they were both taken over by weeds, either the ones in the seed mix or ones that came from the surrounding area. The late cereal seed mix looked more similar to the reference condition, but it wasn't quite there yet. Even after 25 years, it hadn't really approached what we consider restoration success in terms of its fidelity to the reference plant community. But the idea is that the main outcome of this was an understanding that those founder effects are really important. So what you put in the site and what you create in the terms of a plant community really shifts the long-term trajectory of the overall system. We did another study at this same site, a different study, one that was put in 1977, where they were interested in looking at different organic amendments as a way to do revegetation and mining disturbances. So at the time, organic amendments were regarded as something that was probably going to have to be used to revegetate these areas because the substrate that was left over after mining had very low organic matter. So this study was put in using different combinations of municipal biosolids mixed with wood waste. And then they had two different types of soil substrates that they applied these to. One was a crappy soil that was simply the replaced subsoil, and the other was a good soil that had replaced topsoil. So into this crappy soil or good soil, they mixed these organic amendments at these different levels, seeded them with the same seed mix, and then walked away. And we came back some 25, 30 years later to see what the results would be in the plant community. What we found was not what we would have expected, but on the subsoil, the organic amendments reduced species diversity and resulted in a dominance by perennial grasses. That's actually what we did expect in that situation. What we didn't expect was that on the topsoil treatments, the organic amendments, where we're adding even more organic matter, decrease the perennial grasses and increase the shrub dominance, which is more what we would want in restoration in this shrub-dominated ecosystem. So when you add more organic matter to the topsoil in this situation, it resulted in the plant community going towards more like the reference condition. That's not what we would have expected. I'm not sure why that happened. We speculate that it might have something to do with the biosolids and the level of metals that were in those biosolids, and the mixing of those high metal biosolids with the topsoil that might have bound some of those metals. But we were never able to follow that up to really discern that. The main point here, again, is that those initial conditions really have a big effect on the successional trajectory of the plant community. So that was a fairly important finding from this study. So another research area, this one being led by a Ph.D. student in the lab, Ryan Busby, some years ago. Ryan looked at below ground community assembly as it relates to plant invasion. So Ryan went to 3 different sites in Colorado, Utah, and Wyoming, and collected rhizosphere soil from either native sagebrush shrubs or cheatgrass seedlings growing amongst those sagebrush plants. And he brought that rhizosphere soil back to the lab, and he quantified the arbuscular mycorrhizal fungi that were in those 2 different soil types. He did that by extracting those mycorrhizae, culturing them, and identifying them using microscopy and staining. What Ryan found was that the AMF communities differed rather substantially between the native sagebrush and the invasive cheatgrass. So there were some mycorrhizae taxa that only occurred in one or the other of those 2 rhizospheres. And the ones where there was commonality between the cheatgrass and the sagebrush, there were some differences in their abundance, at least. So fairly different effects of those two plants on the arbuscular mycorrhizal community. And so one way to interpret that is that the soil community assembly is really an emergent property of the invasion of that cheatgrass. The invasive cheatgrass changes the soil microbiome. Those altered microbiomes might in turn influence what plants that can be recruited in that system in the future. So if the cheatgrass is changing the mycorrhizal community, that might change the ability of native plants to be able to invade that or to come into that system after the invasion. So an emergent property based on that feedback loop. So Ryan continued on with these studies, and he did another study where he examined both the plant response to AMF and AMF response to plants. And that was something that was fairly unique at the time. People have done a lot of studies looking at the impact of mycorrhizal fungi on plants, but Ryan combined that by looking at the impact of the plants on the mycorrhizal fungi, which was pretty cool. And he did this by looking at 17 different early and mid-soil native plant species using a homogeneous AMF community. So he measured the plant responsiveness to field soil inoculation with and without AMF. So he autoclaved the soil multiple times to kill off the AMF, compared that soil with one containing the mycorrhizae. And then he measured the mycorrhizal density on roots of plants in a bioassay using a common plant species that had been trained by the individual host species that were used in the responsiveness study. So what Ryan found is that plant species are highly variable in their interactions with AMF. So the visual evidence here is pretty striking. You have ambrosia either with mycorrhizae or without mycorrhizae. And in this case, ambrosia doesn't like to grow with the mycorrhizae in terms of its productivity. So the mycorrhizae here is a parasite on the artemisia. And what you see by looking at all of these different responses of the plants to the mycorrhizae and the mycorrhizae to the plants is that some plants benefit from mycorrhizae, other plants do not benefit from mycorrhizae. Similarly, some mycorrhizae benefit from plants and other mycorrhizae do not benefit from plants. So you can have mutualisms, parasitisms, amensalisms, commensalisms, all being prevalent within a community of plants interacting with mycorrhizal fungi, which makes it very hard to interpret, but also dangerous to also interpret it as always a beneficial organism or beneficial relationship. And one of the main outcomes of that is that the presence of certain mycorrhizal fungi species may have founder effects on the plant community. So by benefiting some plants, not benefiting other, the soil microbial community may be influencing the composition of the plant community. So around the same time, we were working a lot with the mining industry, and we were doing a lot of research on metal toxicity of plants because the mining industry in Colorado creates a lot of conditions where there's high metal content in the soils. They were interested in looking at how to do restoration on these areas that have high metals. And one of the approaches is to use hyperaccumulating plants that suck up a lot of metals out of the soil. And we were doing this work in collaboration with Elizabeth Pilon-Smits in the biology department. It's no longer here. And this was led by Ilan Alford, a Ph.D. student in my lab. Ilan was interested in delving into the potential relationship of these hyperaccumulator plants with the soil microbiome. And she started that by doing a review as part of her dissertation, looking at the relationships between metal hyperaccumulation and soil microorganisms. And what she found in doing this review was that indeed there's very strong relationships, lots of evidence in the literature that rhizosphere microorganisms are really important in regulating the uptake of those metals by the plant. So what we see as a plant trait is really not a trait of the plant, but of the plant microbial combination. The community is creating what we see as a response in the plant, influencing the plant metal uptake. Soil chemistry and biology really co-regulate the observed responses that we see in plants. It's not just the plants acting independently. So those emergent properties of the rhizosphere community are really important for determining plant traits that are valued in restoration. So things like phytoremediation, again, controlled by microbes. Continuing on the same thread, another PhD student in my lab, Jose Valdez-Barrios, back in 2007, was interested in toxicity of white loco weed. You've probably heard of white loco weed. It's a very common plant in the Western United States. It's estimated that it causes somewhere between thousands and tens of thousands of livestock deaths every year in Western US, which I think is more than wolves. But nobody gets that excited about loco weed for some reason, but it's killing thousands or tens of thousands of livestock every year. What we knew at the time was that the toxicity of loco weed is attributed to a compound called swainsonine, which is produced not by the plant, because plants are really crappy chemists, it turns out, but it's produced by a fungal endophyte that lives inside the tissue of the plant. So this fungus, Embolisia, lives in the tissue of the plant, and it produces the swainsonine toxic compound that can be toxic to livestock. So it's providing a defensive mechanism to the plant and resulting in differences in plant community composition based on that. What intrigued Jose and myself was the fact that loco weed is in the legume family. Legumes fix nitrogen, most of them anyway. Nobody knew for sure whether white loco weed did or not. But since swansonine is a nitrogen-based defensive compound, we figured that there might be a relationship between not only the fungi, but potentially rhizobium and root nodules, increasing nitrogen supply to the plant, increasing swansonine production by the Embolisia fungi. And so Jose did a study to determine if swansonine production could be influenced by nitrogen fixation by bacteria and root nodules. And so he set up an experiment where the plants were either with rhizobium or without rhizobium, and with Embolisia or without Embolisia. So we knocked out Embolisia by using a fungicide, and we added rhizobium using cultures of rhizobium. And then we measured swansonine production and nitrogen fixation in the plants. It turns out that the plants did fix nitrogen. They formed effective nodules. So that was the first report in the literature of nitrogen fixation by this species. Also, nodulation resulted in increased plant swansonine concentration. So that link was confirmed. So when Jose knocked out the fungi, it also reduced the swansonine concentration. So here we have a system that's a tripartite mutualism, where the plants, bacteria, and fungi are all interacting together in complex ways that influence herbivory and plant community structure. So another emergent property arising from interactions between plants and microbes. So another graduate student, Shabana Hussain, more recently was interested in a similar thing, and she was interested in how arbuscular mycorrhizal fungi might be related in their function to soil bacteria. So we tend to think of mycorrhizal fungi as having this symbiotic association with plants, but we tend to ignore the fact that those mycorrhizal fungi also have symbiotic relationships with bacteria. And so it's not just a simple system where there's one-on-one, it's multiple species of fungi potentially interacting with multiple species of bacteria, causing what we seem to think of as simple interactions when they're really quite complex. So Shabana did a literature review to start off her work looking at the role of bacteria symbionts with AMF, and sure enough, there's lots of evidence that that is indeed the case, that there's a lot of mutualistic interactions. And she continued her study with a field study. So she collected soil from a long-term research project in Nebraska, where researchers there had established a restoration study using low diversity seed mix, medium diversity seed mix, and a high diversity seed mix. That resulted in a plant community that either had low diversity, medium diversity, or high diversity. Shabana went and collected soils and did a microbial network analysis in collaboration with Pankaj over in the soil science department. And what Shabana found when she did this network community analysis of the bacteria and fungi was that there were very much positive interactions amongst bacteria and fungi that increased with plant diversity. So this is a fairly complex figure that shows a really simple idea. If you look at the purple lines versus the blue lines, or pink versus blue, it's either a positive interaction between that bacteria and that fungi, or bacteria and bacteria, or there's also AMF in here as well. But what you see by mapping out these interactive networks between bacteria and fungi in the soil is that when you have more plant diversity, things change rather dramatically in terms of those interactions. You have a system with not much positive interactions and a lot of bacteria, to a system where fungi become more dominant and positive interactions become the norm, as opposed to negative interactions or no interactions. So as you increase plant diversity, you increase those positive interactions, those facilitative interactions between bacteria and fungi, which probably in turn has a lot to do with increasing diversity within the plant community. So all related. So more diverse plant communities lead to those positive interactions. So summing it all up, I kind of cherry picked some studies, but one of the points I want to make is that there's a lot of other studies in these areas that my lab has conducted that sort of contribute to this, but I just picked some of them that sort of illustrate the main points. So the soil nutrient cycling, we know that shifting nutrient cycling can shift competitive rules within plant communities, that shifts the community assembly. In terms of invasion resistance, we know that invasion changes the soil community, which changes recruitment filters and can act as a feedback loop within plant communities. Above and below ground coupling, plant microbial shifts during community assembly occur in parallel, and how they occur is yet to be determined in the details, but we know that those shifts occur in parallel. It's not one driving the other. Plant community diversity is a really important driver of restoration success, probably via emergent properties that are related to what's happening in the rhizosphere that we talked about. And those long-term legacies are really important. So what we do in restoration can have really long-term effects. So when we're thinking about what we're going to do in restoration, we should be thinking very hard about what the long-term implications of those are, whether that be a seed mix or some sort of soil amendment or other treatments. Those can have very long-term effects. So I said I would come back to this policy question. So if we're going to move to more of a process-based approach to restoration, how do we go about developing policies that can focus on that? I was pondering this question when last week I just happened to pick up a copy of Restoration Ecology. And in last week's version of Restoration Ecology, Jens Svenning published an article on this exact topic, arguing that we can redefine how we think about restoration to a more process-based approach. And Jens goes into talking about how that relates to how we think about monitoring and regulating restoration projects. So these ideas are starting to take root, and maybe in the future we'll get to a point where we can actually regulate and implement restoration projects that are more in line with process-based as opposed to species or form-based. So lots of acknowledgments. Again, I kind of cherry-picked different topics, but the ones I picked had a lot of collaborators, both here at CSU, several postdocs, couple lab managers, lots of grad students, too many undergrads to count, and a lot of different funding agencies. So I will stop there, and I think we have a minute or two for questions. Or a few extra minutes to go. Yeah, Chuck.
So, Mark, when you started this, there was no such thing as climate change,
True.
and now
There still isn't, Chuck.
there's right. Depending on who you ask, there's still some people are hard to teach. Thinking about, there's so many things you've been involved in, so many projects and so many different aspects of this. If you were going to sort of lay out a plan for future work or think back to what you would have done, knowing more about where we are in terms of how climate has changed, how would that maybe have informed some of these studies? I mean, you focused on nutrients, you didn't speak too much about soil moisture, but perhaps that might have been an area where you dug in a little bit more. I'm just kind of curious, when you think back over the decades, what might have changed?
I think what's changed for me, but also for everybody else in the restoration field, I think it's the importance of biodiversity. And if we're moving into a future where there's great uncertainty about climate, we probably should be trying to restore as much biodiversity as we can and then let nature sort it out. That's what I call the shotgun approach. When we talk about putting seed mixes together, my approach these days is use everything. Not only every species, but every genotype of that species, and let nature sort it out, because we don't know what the future is going to bring. Could be hotter, could be drier, could be wetter. But by having that diversity, at least we're setting up the site for long-term possible vegetation. I don't know if that got to your question, Chuck.
Yeah, no, that's great. Thanks.
Lee?
So after fires, and Chuck can speak to this more than I can, but you get this pulse of nitrogen coming out. And so does that pulse of nitrogen then result in more invasives than you would have if you didn't have that pulse of nitrogen? Or is the invasives just coming because they like to fill in a vacuum?
Yeah, that's a good question, and it brings up an important point, and that is that not all invasives are nitrophilic. Not all invasives like high nitrogen. When we talk about the invasives that like that high nitrogen, we're usually talking about annual plants. So annual invasives like cheatgrass. That's the classic example of a nitrophilic species. And yes, after fires, we see cheatgrass coming in, probably somewhat related to that initial flush of nitrogen, creates just great conditions for cheatgrass to flourish and put down a lot of seeds for the future. But yeah, annual invasives for sure, but also native invasives. Native weedy species are what we see coming into a lot of post-burn areas, but they're just offset by so many non-natives that are in the seed bank.
I guess if you could magically not have a flush of nitrogen after a fire, would the pattern of invasives look similar or different?
I think it would look different. It would probably look potentially more bare ground, because you not only would not have the invasive annuals, but you wouldn't have the native annuals coming in either.
Right.
So I think what we need are more native annuals and less non-native annuals to take advantage of that flush of nitrogen.
There's a question online, but I can't tell if it's two-part questions or two questions.
All right. I'll interpret it however.
Okay. Truman Young says, nice talk. It seems that your first study showed profound but transitory effects of N and see in these plant communities? At the end, what were the most interesting differences? And then also, diversity experiments in plant communities tend to show saturation of benefits at diversity levels that some might consider relatively low. Do you have any sense of this for soil microbes?
So I'll answer the first one first. In terms of legacy impacts, what we saw in most of our studies was there's a longer legacy of the nitrogen removal, the sucrose addition, because that results in dominance of perennials that have a longer signature on the landscape. Those nitrogen effects were more transient because they resulted in the dominance of annual species that went away quicker after that nitrogen addition stopped. So the legacy of removing nitrogen is longer than the legacy of adding nitrogen. In terms of the second question, the sweet spot for diversity, yes, I've seen lots of studies that agree with that, Truman, that certainly there is an upper limit to how much diversity you might want to have. My comment on that was more about the uncertainty of the future in terms of putting seed mixes together. I would not expect all of the species we put in a really diverse mix to ever establish, but at least giving them a chance gives us a chance to have vegetation there in the future. In terms of the microbial community, I don't think anybody really knows. There's lots of new tools that can analyze the soil microbial community better than we did at the time that we did this study. So hopefully in the future we'll have that answer, but I don't think we're there yet. Eric.
In terms of species, yes, like along with taxonomic diversity, if you're looking at the functional traits of species within those mixes, or like any other traits that might be beneficial for different areas or for future uncertainty.
Yeah, we could think about diversity in a lot of different ways, but I think those trait diversities are important. I think that mycorrhizal study that Ryan Busby did, with the positive and negative impacts on the mycorrhizae and the plants is a good example of, plants with different traits may result in differences in the soil microbial community that could have impacts on future assembly within that plant community. So yes, thinking about it from a trait perspective, I think makes sense as well.
Okay, let's thank Mark again for sharing.
I think there's 17 people online.
Wow.
Yeah.
That was super fun to see, like, mix of all your work put together. Yeah, some of it. Yeah. That'd be a mess. Like, what did I learn? Cool.
Take a job.
Yeah. Cool. Well, thanks for